more comfortable if you're a little bit farther up, so you can move around a little bit. I appreciate that. Let's take a moment and introduce Dr. Schoichet to you. You know, we're so often impressed by some research that's being done somewhere in the States or some leader in the academic field, research field. Molly is a Canadian and a UT professor, and I'm going to read just a little bit about her because she's done so many different things. This is a little citation from the Trump Star. This is written about the time she received the significant UNESCO award. So here's a little bit about Molly. Collaboration is the key to her success. UT professor studies polymers that can repair spinal cords and brains. Now, how important is that in our world? Broken pathways and how to reconnect them are what Molly Schoichet spends much of her exceptionally long work days thinking about. The University of Toronto chemical engineering and biomaterial professor studies polymers that can be injected into the body to repair damaged spinal cords and stroke-besieged brains. For her pioneering research in this area, Schoichet has been honored with a Woman in Science Award from UNESCO and the Loyal Foundation. The International Prize provides a platform to discuss another broken pathway, the advance of women in science, technology, engineering, and math, otherwise known as STEM, STEM, of course, and all the careers that are involved in that area, where the gender gap truly yawns. So I could say an awful lot more about her. She's written hundreds of papers. She's done presentations all over the place. So without further ado, I'm going to turn things over to Molly.
Okay, thank you. Well, this is a little bit more of an intimate group. I know I'll come down here and I'll go up there periodically just to change some things around. It's really a privilege to be here. I want to tell you that my chemistry high school teacher was fantastic and had a huge influence on me in my career. And so I was searching around the internet, and it's not such a great photo of him, but this is Alf Allen, and I went to the Toronto French School, and he was really just a really inspiring chemistry teacher. My brother, who's 2 years older, I have two actually older brothers, but one 2 years older than I am, also went to the Toronto French School, was also taught by Alf Allen, and also pursued a career in chemistry. And so I salute you guys. You guys have a huge influence on our lives and careers. I teach chemistry to engineering students at the University of Toronto, and I can always tell who has a really strong foundation and who can, who's had that inspiring chemistry teacher. Other people have been hugely influential in my life. So I went on to study chemistry at the Massachusetts Institute of Technology, and it was in one of our advanced organic chemistry labs that we did polymer. And one of my lab mates said to me, Molly, close your eyes and stir this solution. And so it was a liquid, and as I was stirring it, it was polymerizing and constantly. And so not that I normally recommend that people close their eyes when they're doing chemistry experiments, but for that small period of time, I went and it was stirring, it was easy, and then all of a sudden I couldn't stir. And that was really inspiring to me because I thought, wow, this is so exciting. This is a chemical reaction that I can actually see. And I went on to take a series of polymer courses at MIT, and this is a picture of Ed Merrill, who's a chemical engineering professor, or was one, now it's emeritus. And he was hugely influential because kind of encouraged and continued to inspire me in polymer science. I went on to pursue my PhD in polymer science and engineering at the University of Massachusetts in Amherst. And Tom McCarthy was my PhD supervisor, and this is what he looked like then, so that's why I chose, the internet's great, right? You can see what people used to look like. And so he also fantastic to me because he allowed me to kind of pursue the things that I was interested in. And when I was at MIT, I was also pre-med. I grew up with a very strong role model in my mother. Growing up, actually, honestly, this is going to sound ridiculous, but I can remember being very young, maybe 3 or 5 years old, saying, I'm not going to get married until I'm 35 and my career is well established. Actually, I got married at 28, and I felt so far ahead of my deadlines. I was really making faster progress. But anyways, Tom was a great supervisor because he could have cared less about biology and medical applications, but allowed me the freedom to explore during my PhD, and that led me to start my career in industry at a company called Cytotherapeutics. And cytotherapeutics, we were looking at encapsulated cell therapy. So this is this idea where you're taking cells and they are producing the drugs that we need to overcome really significant diseases like Parkinson's disease or diabetes. And so instead of delivering drugs, we're delivering the cells that will produce the drugs. And the drugs are protein drugs. And it just turned out, that's really lucky. I was really started to work in this field of regenerative medicine as the field was evolving. And so when I came to the University of Toronto to start my academic career, it's a little bit hard to see, but Michael Sefton was working in this field. He was the person that attracted me back into academia because I was really excited about coming back home, coming back to Toronto, but also continued to work in this field that I knew was at the cutting edge of research, at the cutting edge of science. And so he's been a huge mentor, as has Mitch Winnick, who is a professor in chemistry as well. And then I showed this. This is from the L'Oreal UNESCO Awards because it's a UNESCO award sponsored by L'Oreal. You can imagine that it's got the power of marketing behind it. And in Paris, there were 4 of us women from around the world who got this award. So me from North America, and there were these big banner ads, like you'd walk down the streets of Paris. So it was pretty cool when I saw my own. And you can't see my mom here or me, but we're standing there. But I show you this not because it's a great picture of me, because it's a great picture of me, but because what it says is la science a besoin des femmes, right? And science needs women. And I think you all have such a tremendous role in terms of inspiring our youth to pursue careers in science and engineering, but encouraging our full population to pursue those careers. And we are trying to tackle really big problems, and we can't depend on just half the population to solve that. And so, that's why I think it's really important to encourage both men and women to pursue careers in science. So that's just a little bit about me and sort of a little bit of my journey and how I got here. I've now been working at the University of Toronto for 20 years, which seems, I'm teaching, the students I'm teaching this year were born when I started to teach, so that's when you start to feel old. But it's been a great ride. I love this idea of curiosity-driven research. I love the idea of trying to come up with solutions to really big problems. And really what we use is chemistry and engineering tools to solve problems in biology and medicine, so highly interdisciplinary. And what I thought I would do is tell you a little bit about our research, how we're using chemistry and engineering to overcome really big problems, such as those in cancer. I'll tell you a little bit about our research in blindness and in spinal cord injury, and really how we're using some innovations and new materials. So these are the innovations in polymer science, but they're inspired by the problems in biology and medicine. I'll tell you a little funny story that my son Emerson, who's now 17, when he was 9 years old, he said, Mom, when I grow up, I want to be a scientist. And I said, I'm a scientist. And he said, when I grow up, I want to invent things. And I said, I invent things. And he said, yeah, not like you, Mom. So it was really funny. This is a short movie that I thought I would show you. And it really describes some of our research in cancer. And then I'll show you some of the science behind the movie. But it was made by Corinne Ho, who did her PhD in my lab in chemical engineering, and then decided that she was really enamored with the communication of science. And so after getting her PhD, she went and got a master's in biomedical communications, also at the University of Toronto. And I was really excited that she wanted to create a small movie, a short movie, so it's only a couple minutes long, really describing the challenges faced.
Chemotherapy can cause severe side effects because it is nonspecific and damages healthy cells as well as cancer cells. To manage systemic toxicity, doctors must limit drug doses, making aggressive cases of metastatic cancer difficult to treat. At the Schoichet lab, our goal is to deliver chemotherapeutic drugs using a targeted system to make treatment safer and more effective. We developed a biodegradable polymer that self-assembles in water. This process yields drug-loaded nanoparticles. Our nanoparticles are so small that they are 1,000 times thinner than the width of a human hair. At this size, nanoparticles have special properties that allow them to target solid tumors. By controlling their features, we can optimize their properties for drug delivery. Our system targets primary tumors and metastases using size-based and molecular targeting. Blood circulation distributes nanoparticles throughout the body to anywhere that cancer cells may be. Our strategy then uses size-based targeting to guide the drug-loaded nanoparticles to tumor sites. Normal blood vessel architecture is disrupted in solid tumors. Tumor blood vessels are disorganized and leaky. This allows our nanoparticles to pass through and selectively accumulate in tumor tissue based on size. Preclinical studies using our nanoparticles show improved drug tolerability in animal models. Compared to the free drug alone, our nanoparticles also keep drugs in circulation longer. This leads to greater tumor retention and extends exposure of cancer cells to the active drug. To further improve our nanoparticles, we are using antibodies for molecular binding to cancer cell surface markers. To attach antibodies to our system, we are using click chemistry. This reaction preserves the strength and specificity of antibody binding, enabling target cell recognition. Binding improves uptake, bringing the drug inside the cell, which is the most common site of action. Attaching antibodies to our system enables selective binding and uptake by live target cells in culture. By controlling antibody density, we can adjust nanoparticle binding strength predictively. We are continually testing and refining our nanoparticles to improve stability, versatility, and targeting. This research will be used to validate our system as an alternative to conventional chemotherapy. Our expertise in targeted drug delivery enables our team to accelerate towards our goal of bringing this technology to the clinic. By optimizing our nanoparticles to target tumors on both tissue and cellular levels, our system has the potential to enhance cancer treatment and improve patient care.
Okay, so maybe what I can do now is just tell you a little bit more about the science behind that project. But what you can see, or what you saw, hopefully, was the science, actually the data was incorporated into the movie. So what we know in cancer is that we have these really potent chemotherapeutics. And the problem isn't that the chemotherapeutics aren't strong enough to kill the cancer cells, except they kill all rapidly dividing cells. And so there's an area of research around this idea that we can enhance tumor targeting by taking advantage of the hyperpermeable or leaky vasculature that surrounds some cancer tissues. So the idea then is that small drug molecules, like shown here on as these little yellow dots, can permeate. So, drug that's taken intravenously or orally gets throughout the body by the vascular system. And so a small drug can permeate across both healthy tissue and disease vasculature into the tissue. Whereas if you put that same chemotherapeutic, if you packaged it into a little nanomine cell, that even though this is small, it's still too big to traverse healthy vasculature, but it can now traverse disease blood vessels. And so taking advantage of that greater permeability in the disease tissue, that we can selectively accumulate chemotherapeutics at the tumor by this hyperpermeable vasculature. And so that's just this idea of taking advantage of size. And then once it's there, you can promote greater selectivity and targeting to the cancer cells by that targeted interaction. So these little Ys representing antibodies that will selectively bind to the cancer cells. So hopefully, what you can see even in that small video, in that short video, and in this research, is that we're already combining like innovations in chemistry to solve these problems in biology and medicine. And so what we did then is we synthesized a polymer that would self-assemble into these nanomicelles. And in fact, yesterday I just talked to my chemistry class at the UT, and I was teaching them about self-forming micelles in solution. And the same thing is true here with the polymers. They're amphiphilic, and in an aqueous solution, they'll form these nanomicelles. And we put polyethylene glycol, so we specifically designed the polymers with polyethylene glycol on the outside, because polyethylene glycol has been shown to limit protein absorption and cell interaction to materials. And then we also designed these to allow facile chemical modification with the antibodies to allow that directed targeting. So these are the polymers that we synthesize. So we actually go all the way from the small molecule to the polymer. Lactide is polylactide and polyglycolide are very common degradable polymers found in sutures. So we take advantage of a material that's well known, combine it with this trimethylene carbonate, which has a carboxylic acid group, and that allows us to graft on polyethylene glycol. And then in the film, you heard about click chemistry. And so we designed our polymers with this furan group that enables us to do certain chemistry where you have two functional groups that will only react with each other. And they will react with each other in water, in an aqueous-based solution. And that's always important to us because these polymers will dissolve in organic solvents, whereas in an aqueous solution, we can form the nanomicelles and they'll be stable, and then we can do water-based chemistry. And then, of course, if we're working with antibodies or proteins, those will also degrade in organic solvents. So it's nice to keep everything in water. So what happens is exactly what would happen with a soap micelle. A soap micelle, will dissolve dirt or grease by selectively like a hydrophobic interaction and selectively taking up that grease into the inside of the micelle. So the way we incorporate a hydrophobic drug, such as docetaxel, which is commonly used in breast cancer treatments, is we just take advantage of the fact that this is hydrophobic. The inside or the core of our nanomicelles is also hydrophobic. So during that self-assembly, it will become incorporated into the inside of the nanomicelles. And then these little gray hairy lines are meant to represent polyethylene glycol, which helps stabilize those polymer micelles. So basically then, as we start off with an organic solvent in which our polymer is dissolved and our hydrophobic drug is dissolved, we mix that with an aqueous pH buffered solution, and we put that into a dialysis bag, which is just like a tea bag. So it's a material that will allow the exchange of solvents. And so water will come in, our organic solvent will leave, and we're left with these nanomicelles. And you can actually see with your own eye the difference in the size, depending on how we've changed the solvent here. We can change the size of the nanomicelle here. So this is a completely clear solution, not because there are no micelles in it, but it's just because the micelles are so small that they're not refracting the light. Whereas here, as we've changed the concentration of the borate buffer, or not use any aqueous solution at all, they get, this is obviously darker than this, and this is increasingly more opaque, and that's just a reflection of the size of the nanomicelles. So I would love to tell people about what we just done in research, and this is work that Logie did in the lab, and we just published this about a couple months ago, and I thought this was really clever and really innovative. One of the big challenges we have with drug delivery in these nanomicelles is loading. We end up delivering more polymer than we do drug. And so there's a big challenge in the field. How do you get more drug to sequester inside of those nanomicelles? So we take advantage of that hydrophobic interaction. What can we do any better? And so what other people have done in the field is they said, well, let's just make that hydrophobic core more hydrophobic, right? So it'll have a stronger interaction, and that works okay. So we did the same thing, or Jen did the same thing, where she had just a benzyl group here that makes our hydrophobic backbone more hydrophobic. And then the other thing she did is what other people had also done is she took the drug itself, docetaxel, and she incorporated that into the polymer chain. And then docetaxel will form like pi stacking interactions with itself. And so that also works pretty well. But I think what was really clever and really made a significant difference is she took advantage of a really she took advantage of a really well-known biological interaction. So the way docetaxel works is it binds to what's called the taxol binding peptide in the cell. And she took that well-known biochemical interaction and decided, I'm going to use chemistry and incorporate that taxol binding peptide into my polymer chain. And with the hypothesis that the docetaxel then would chemically or biochemically interact with the taxol binding peptide, and we could increase loading. And that's what, so it's really hard to see, I apologize for that, but that's what we're showing here. So we're looking at the amount of docetaxel loaded, and you can see that modified polymer, we're only at about 10% loading. When we do these hydrophobic modifications, either with the benzene ring or the docetaxel, we go up to about 20%. But with the taxol binding protein, we go up to close to 50%. And with the scrambled peptide, so it's not just that it's being made more hydrophobic, there really is a specific interaction. And if you take the same amino acids, but just scramble the sequence of those amino acids, again, you go down to sort of generic levels of increased loading, but not nearly as much. So this is really exciting to us because it overcomes what's been a really big challenge in the field, and now gives us the opportunity to explore this in cell-based assays in the lab, and then also in animal models of disease. So Corinne Ho, who made that movie that you saw, when she was a PhD student, she took those nanomicelles and tested them in animal models of disease. So these are, this is a breast cancer model. It's an orthotopic model, which means we're growing human cancer cells in in an area in a mouse that would be similar to where they would grow in us in the breast tissue. So it's called the mammary fat pad. And these are immunocompromised mice because we all know that if you get a splinter or anything foreign, the body's going to try to remove it and get rid of it. So we have to, now that we're putting human cells into mice, they have to be, we don't want those human cells to be destroyed or eliminated by the immune system. So we use immunocompromised mice, and that allows the tumor cells to grow. And then we can look for things like selective accumulation in the tumor cell, because that was the hypothesis. So I have to tell you that we spend all this time designing the polymers, synthesizing them. We have a hypothesis, we're really excited. And then right before we do an animal study is when we're most excited, because that's kind of the moment of truth. Will things like this decade-long of polymer synthesis actually be useful? So we were really excited when we thought that there was some utility. So what you're seeing here in this graph is the delivery of 3 dose attacks. So the way the drug would normally be delivered intravenously versus delivering the same drug, but in our polymeric nanomicelles or nanoparticles. And what you can see is this is just the dose remaining in circulation. And you might say, why do you care about circulation? And the idea is that the more times that drug circulates in the blood, it gives us more opportunity to accumulate at the tumor tissue. So we were really excited to see that the drug is accumulating significantly longer when we deliver it in our nanomicelle versus the free drug alone. But look at the data, it's a log scale, right? We started at 100%. So in a very short time, we get down to 10% of our dose. So while we're showing a significant improvement, and it really is a significant improvement, we still know that there's a long way to go in terms of improving this strategy. The other thing we looked at is we looked at all the organs and we said, well, where is the drug accumulating? Is it actually accumulating in the tumor? Or is it accumulating in some of our organs, the natural filtration organs in the body? And what we see is in all these liver, kidney, spleen, not like the heart and lung or filtration units, but we see in all of them it goes down over time. Only in the tumor do we see that the accumulation of the drug continues over an 8-hour period of time. So this is good. It's hopeful. It gives us the hope that this is worth pursuing and we should continue to do the chemistry. We're currently working on efficacy studies because this just says, yeah, it accumulates, but does it do anything? And so that's where we are right now, looking at some of our improved formulations, the improved chemistry, and looking to see if it will actually do anything useful. So now what I thought I'd do is change completely, and so for the people who came in who really just missed the first chapter, but chapter 2 and 3 is completely different than chapter 1. And this is this area of regenerative medicine. And in regenerative medicine, what we're doing is we're thinking about cell transplantation and cell stimulation. So what's really exciting in the brain, for example, and in areas of stroke, is that we all have resident stem cells in our brains. And the challenge is, how do we stimulate those to promote tissue regeneration and tissue repair and functional repair? And that's an area that we're actively pursuing in stroke and also spinal cord injury. And the other area that we're pursuing is stem cell stimulation. So if we cannot stimulate the stem cells that are resident in our bodies, can we then instead transplant cells to overcome that loss of function? And that's an area that we're pursuing in spinal cord injury, stroke, and blindness. And so those are the stories that I will do today. And so because it was early in the morning, I thought lots of movies would be good. So this is another movie that was made a couple of years ago on some of our spinal cord injury research, and it was made by another biomedical communications student, Lee.
In a spinal cord injury, the initial trauma is just the beginning of a devastating condition. Damaged axons are blocked by glial scars and inflammatory cysts and start to degenerate in a hostile environment. Some neurons with severed axons may, however, survive. If these injured axons could be made to regrow to their innervating targets, a certain level of functional recovery might be possible. During development, and whenever neurons must grow later in life, this growth occurs at a highly motile axon tip known as the growth cone. According to recent axon-guided studies, the cone's pathfinding is guided by a number of mechanisms, including both attraction to and repulsion from the extracellular matrix, and long-range chemoattraction, mediated by various neurotrophic factors secreted from target cells. In an attempt to simulate these axon guidance mechanisms, we are developing a tissue-engineered scaffold system. A concentration gradient of nerve growth factor, or NGF, is immobilized in bioactive 3D gel. The gradient slope can be controlled. Lab-grown neural cells are placed on the matrix. After the cells into the matrix, we use directional data analysis theory to investigate pattern output. Within the gel, a coordinate system is defined with its origin at the center of the cell body. Normalized point set data for the positions of the neurite tips can be obtained from the coordinate system. These lines represent the location of neurite tips and show their angles relative to the direction of the gradient. Numerous samples from each gradient are statistically analyzed to test for directional bias, to see which NGF gradient slopes will most effectively elicit axon guidance. Our experimental results show that we can achieve directed neurite output by varying the slope of the NGF gradient. We believe this system can be combined with other strategies to develop a regenerative therapy to guide axons across the gap, potentially allowing severed axons to regenerate and reinnervate their targets.
So one of the big challenges with spinal cord injury, and really with all the big diseases we're going after, is that there's very little for the patients. So in spinal cord injury, there's one drug that's being approved, methylprednisolone, and it's really debated whether it's actually anything at all. So we have rehabilitation, which is great, but it's a really big challenge to the community, and it's why, something that inspires us as well. So I had the opportunity to meet Rick Hansen a couple of years ago, and he's actually been, a huge spokesman for spinal cord injury and really a Canadian hero for taking, a devastating, traumatic injury and turning it into something really positive for Canadians. So what we have in spinal cord injury, Christopher Reeve was also a huge spokesman for spinal cord injury. It takes the most active in our population without warning, and renders them disabled. And so Christopher Reeve, while he's now passed away, was a huge spokesman for spinal cord injury. What happens with spinal cord injury is you basically lose function below the site of injury. So with Rick Hansen, his injury was further down. And so he doesn't have the use of his legs, but he has the use of his upper body. With Christopher Reeve, he fell off of a horse and he injured his spinal cord really just kind of like under his head, really in the cervical area. And that's why he lost function that's why he lost function of, really his upper body and his lower body. So it's a completely, a very complex injury. I think of it as you have a big cable of wires that have now been severed, and we have no idea how to reconnect them. And, simply just throwing in a bunch of wires isn't going to work. To figure out how to reconnect those wires. And it's a huge challenge because the brain and spinal cord obviously are the control centers for our entire body, and so it's a huge challenge. And it's also, it usually affects the most active in our population. So usually those between 16 and 45, and it's there's been huge advances made just in safety, so that limits the number of people with spinal cord injury, but really still a huge problem. So, and I'm up here so because I'm going to show you another movie, but it's sort of a real data movie. We've been working very closely with Charles Tatter and Michael Healings, who are both neurosurgeons at Toronto Western Hospital. And in one of our research areas, we thought, what we're going to do, so if blood or guts, makes you squeamish, then you be more. So it'll only be up there for like 30 seconds. But we basically took a spinal cord and cut it completely in half. This is what it would look like then. This is the tissue. And then what we did is we inserted a tube, a chitosan tube that we had designed in our lab, and we've incorporated a series of different strategies into the tube as a way to promote tissue repair. So we incorporated stem cells, and we incorporated this small molecule viral cyclic AMP. And this is what it looks like in the animal model. So after we put sort of like the best things that we thought would be useful to promote repair, then we waited a certain amount of time. And what I think is so cool from an engineering perspective is the tube itself. So we designed the tube with all these really cool features that we could have local and controlled release of our small molecule over time. It's made of a polysaccharide, so a polymer chitosan that's naturally derived from chitin, which is from crustaceans and their shells. And the cool thing, I don't know if you guys can see this, but the tube is transparent. Right, so this is the tube, and this is the tissue that's going in. And now we removed the tube, and you can just see the tissue. So we did see tissue regeneration, and from an engineering perspective, it was just kind of cool that you can just look at it and see. I mean, of course, you have to open the animal up, but so these are two videos. I want to let you guys know that we do animal studies, but we do everything we can before we do an animal study, and that's because very obviously the ethical treatment of animals is important to us, and also practically these studies take a long time and they cost a lot of money, and they're very difficult to interpret. But I show you these two studies, or this one study. This is a control rat, and this rat had an injury that makes it is similar to a paraplegic injury, and you can see that the rat is was dragging her hind legs. This rat, on the other hand, was treated with one of our strategies and is not walking perfectly, but you can see that she is using her hind limbs, not all the time, but clearly much better functional repair. So I want to tell you a little bit of a disclaimer that this was our best rat. And so I show you our best rat, not because I'm trying to skew your mind and show you just the best data, but to show you the promise of regenerative medicine, and that there really is great promise with it, but of course it needs additional research. So more recently, we've been looking at less invasive strategies to promote repair, and one of those is with this hydrogel, polyuronic or methyl cellulose. So polyuronic or hyaluronic acid, maybe you've heard of? Yep. So it's a really common material. It's a polysaccharide. People will inject it to get rid of wrinkles. So it's used a lot in cosmetic surgery. It's also being used around cartilage repair. Methyl cellulose is also a very common material. It's used in surgical applications as well. It's also used as a coating for on tablets for drugs. And we combined these two materials and we saw really interesting things. So you can tell I like movies. This is actually a little movie that Mike Cook is a close up in the lab made, so there's no sound with this one. And all you're going to see is we've taken our hyaluronate methylcellulose hydrogel and colored it blue. And really all I'm showing you is that this material is a gel in our syringe, but it's shear fitting, so you apply a little pressure and it flows out. And here, what I'm showing and what I'm emphasizing here is the size of the needle relative to the size of the penny. So you're going to see the blue gel coming through the needle. The size of the gel really and this blue blob, it doesn't matter so much. I love this video because of course we don't have a penny. I gave a seminar last week in Seattle. So in the US, and I showed them this video and I said, you know, we all know that it costs more to produce a penny than a penny is worth. And we in Canada are so smart, we got rid of our pennies. So they love that. Because they of course still have pennies. So this is our hydrogel. This is kind of how we imagine polymers as sort of like spaghetti. You know, this is a cross-linked hydrogel. It's loosely cross-linked. The methyl cellulose forms these hydrophobic interactions in aqueous solutions. It's an inverse thermogelic polymer, which just means as you increase the temperature, it forms a gel. You know, most things we think about, if you want to enhance their dissolution, increase the temperature. So that's why it's inverse thermogelling, because as we increase the temperature, it forms a gel. And we can talk about the physical chemistry around that if you want. Polyuronate is shear thinning. So when we mix them, we can still push it through that fine needle, which is what you saw. So we imagine that during injection, that the chains are aligned and they flow. But as soon as it comes out of that needle, it's a gel again. And so one of what we usually do is we think about, okay, what's the problem we're trying to solve? What's our design? What are our design criteria that we have to incorporate into our polymer? And when we initially did this work, we were injecting it into a fluid-filled cavity, and so we needed it to gel very quickly. And so one of our design criteria was vasodilation, and this material certainly met that. And so this is where we first worked with this material, was for local delivery to the injured spinal cord. So a much less invasive strategy than inserting a tube, which is what you just saw. And in this idea then, if we just take drugs and inject them into this fluid-filled cavity, they're slow in this cavity, and so they'll be rapidly dispersed. So we thought, well, why don't we inject those drugs in a gel that will form quickly? It will then allow us to achieve local delivery of the drugs to the injured spinal cord. And so you might be saying, well, why don't you just take advantage of the vascular system? And the reason we don't do that is because of what's called the blood-brain barrier or the blood-spinal cord barrier. And the blood-brain barrier limits the diffusion of most molecules into the brain and into the spinal cord. And so we thought, okay, if we can't go around, if we can't get through the blood-brain barrier, maybe we can circumvent it, maybe we can go around it. And the way to go around it is to deliver the drug to the tissue itself. So the drug, I mean, when we're using the vascular system to get drugs to the brain, the blood vessels just get them there, and then the drug has to diffuse across those blood vessels to get into the brain tissue. So our goal is to get the drug into the brain tissue. So we thought, well, why don't we just deliver it to the brain tissue or to the spinal cord tissue, and then the drugs just diffuse through that. So that's exactly what we did. But anybody who's heard of an epidural or who's had an epidural will know that the last thing you want to do in an epidural is puncture the dura, right? And what we're doing is puncturing the dura, because if we apply our drug on top of the dura, which is this thin membrane here, then our drugs will have to diffuse through the dura, through the what's called the intrathecal space, that fluid-filled space, and then into the tissue, and that won't happen very easily. So we punctured the dura, and we were worried about that. And so when we first did these studies, which were done by Dimpi Gupta, we saw something that was so cool and again unexpected, in that hydrogel seemed to, when we injected it, so it seemed to abut against the hole that we created and promoted healing of the dura. So that then no longer became a problem. And here you can see when we just inject a salt solution in an artificial cerebral spinal fluid, the dura is still punctured. So this is really exciting. And then, whenever we're using biomaterials in medical applications, we're always worried about the inflammatory response. Will that material be accepted by the body or will it be rejected? And here we saw, this is by staining for macrophages, we saw a reduced inflammatory response. So we were really excited about this material, and it's something that it's got these really cool engineering properties, and you'll see it's got really interesting biological properties in terms of wound healing. And I'm going to show you some cell transplantation data that also shows how cool it is. So we've gone on to use this material, reformulate it for different applications. For cell delivery to the brain in applications of stroke, cell delivery to the spinal cord in applications of spinal cord injury, and cell delivery to the retina for applications of blindness. So this will be the last story that I tell you is in terms of how we're using this material to promote cell viability and cell survival after transplantation. And these are two really big challenges that face in cell biology and stem cell transplantation, is that most of the cells that we deliver to the central nervous system die. Okay, so if they're there trying to replace lost function and promote tissue repair, if they're there trying where we want them to integrate, become part of that neural circuitry, just like soldering wires into place, if you have that big cable of wires, they have to survive and they have to integrate in order to overcome loss function. And so that's a big challenge that we're taking. And you're like, okay, so where's the engineering in this? Well, we're designing materials that will promote their survival. So we're designing the vehicles. I think of sometimes our research as like FedEx and drug and cell delivery. What's the package that will get the cells there? What's the package that will get the drugs to where they need to be? So I'll tell you this story that relates to blindness. And, there's a number of diseases that lead to blindness. The ones that we're most focused on are age-related macular degeneration, retinitis pigmentosa, and diabetic retinopathy. And what you can see in all of these diseases, so this is the back of your eye. Anybody who's had an eye exam, you can see the back of your own eye, which is, I think, really cool. But what happens in each of these diseases is a loss of vision. So in macular degeneration, you lose central vision. In retinitis pigmentosa, you lose peripheral vision. And diabetic retinopathy, you have this dapple vision. And so what we know about the eye is that the eye comes up, light comes into the eye and traverses the vitreous and hits the retina, the back of the eye. And so, our pupils, it looks like they're black, but it's just because the retinal pigmented epithelial cells are pigmented and they're black. And so when light comes into the eye, it's these cells here, they're cones, they have this cone shape, and rods, this rod shape. Those are called the photoreceptors. Photolight receptors receives light. They receive the light and they turn those light signals into electrical signals in the brain, enabling vision. And these photoreceptors and these retinal pigment epithelial cells interact intimately to enable vision. So one of my close collaborators, Derek Vanderkoy, he discovered that we all have retinal stem cells in our eyes, and they're found in this area called the ciliary body. And so we all know that we have that black rim that encircles our iris, right? Just outside that black rim is where our retinal stem cells can be found. And those cells can be differentiated into all the cell types that make up the retina, including the photoreceptor cells. And so it's a really exciting time to be working in this field because there's a number of clinical trials going on looking at transplanting cells into the retina. And all of the clinical trials are transplanting these cuboidal cells, these retinal pigment epithelial cells. And I think that makes a huge amount of sense because they are sort of support cells versus functioning cells. But really, the cells that you want to transplant are the photoreceptor cells because those are the cells that enable vision. But as I said, the challenge is that most cells that we transplant die. And so we've been looking at transplanting those cells in an environment that promotes greater survival. And this blue blob is meant to represent the cells and our hydrogel. And so Brian Valius, a former PhD student, what he did is he looked at the conventional way of delivering these stem cells and delivering them in our hydrogel. And what we found is that when we inject them in just a saline solution, they all aggregate and mostly die. Whereas when we inject them in our biomaterial, we get a lovely distribution of cells along Brooks membrane, which is right recommended epithelium. And so that's interesting and that's important because if the cells distribute, they have a greater chance to integrate. And that's exactly what we found. So I'm summarizing, 4 years of research in one slide. But these, what we did is we used green fluorescent cells.
I'm working directly with patients to implement new stem cell therapies firsthand. Canadian researchers are making lives better. Come meet them.
So these are actually being broadcast on Discovery and Discovery Science, which is very exciting to us. I invite you to join us online or on Twitter or on Facebook. And it's really exciting to try and find a way to engage the public more in research. Our research is highly collaborative. Everybody listed here are people that we collaborate mostly with people in Canada and the US. These are the researchers in my lab, all master's, PhD, postdoctoral fellows, technicians. We do have undergraduate students and periodically we'll have high school students working in the lab. Our research is funded primarily by the federal government, so we're grateful to them. And lastly, for all the people who you think may enjoy learning more about this, I'm giving a public lecture on Monday, and so it's free, it's open to the public, it's in downtown Toronto, and I love the title, The Embrace the Geek in You. It wasn't my title, but when my kids were little, I said, I have 2 boys, and I said, if anybody ever calls you a geek, you just say thank you, because I think the geeks of the world are the people who are going to change the world. So with that, thank you very much for your
Any questions before
Excellent presentation. I just had one question with regards to your micelle targeting system. Have you ever tried using the system in a non-immunocompromised animal just to look at whether or not there is potentially an immune response?
Yeah, it's a really good question about what's just going to happen to the nanoparticles. Even in the immunocompromised mice, they are taken out of circulation pretty quickly. So the question is, would they get taken out of circulation even faster in a non-immunocompromised mouse? The reason why we haven't done that is because we wanted to work in a cancer-bearing mouse. So the only way to do that really is to grow mouse cancer cells in a mouse, and then you probably don't have to use an immunocompromised mouse. So that is actually a better way to do the experiment. It just takes a lot longer because you have to wait for those cancers. Those cancers are going to grow slower. The way we do the experiments is we'll grow the cells in the lab, the human breast cancer cells, and then we can inject them into the mouse, and then they'll grow within a month versus over, say, 7 months. But it is a good question, and we have drugs that fail in the lab, and we have drugs that fail in the animal models, and then we have drugs that fail in people. And a big question for the community is, how do you have better animal models of disease? And that is a better model.
I'm assuming that there's no decrease in drug resistance for the cell. I mean, this is a kind of a system that the cell simply goes on the these drug resistant.
Right, so the question is, you know, you might be able to get your drugs more effectively to the cancer cell, but how do you overcome drug resistance? So you're absolutely right, that's not going after that problem. But one of our thoughts is that if we can kill the cancer cells more effectively, then maybe they won't come back and metastasize if we can. But the challenge we're trying to address is how do we more effectively kill the cancer cells, or specifically kill them without causing all the other systemic toxicities. The other issue around resistant cancer cells, really we rely on our collaborators to help us think about, okay, what are the other drug targets that we can incorporate? So we do actually have a collaboration with Masad Dama's lab at McGill, where he's designed some novel siRNAs for gene knockdown. And there, then we kind of look a little bit as kind of like a triple threat. So can we deliver the genes to knock down the cells? Can we use the chemotherapeutics to kill the cells? And can we use our targeting approach? So a multi-pronged kind of perspective is really more of the big picture of how we're trying to achieve that, and you know, and then we're also collaborating with Deb Sidhu at UT thinking about novel targeting ligands, like novel antibodies. So yeah, cancer is a big problem because, and there's no cure for it because it is, it's not one disease, and it is, you know, there's so many different facets.
Sorry, I don't know. Does somebody else have a question? You, that's all.
Thank you for the presentation. It's quite really amazing. I read so many new things. You mentioned that you are working with Dr. Payne, and I'm wondering, any idea when you are going to have cell cancer in Canada?
Yeah, so we have a multidisciplinary collaboration, and actually where I'm going after this is to a meeting of our spinal cord research community and talking about some of the advances that we're all making, but we're all collaborating in terms of cell therapy. And so we have, actually, you know, Toronto is a fantastic place to be working in this field of regenerative medicine because we have world-renowned stem cell biologists like Derek Vanderkoy, with whom I'm collaborating, and Andras Naj and others. So those guys are coming up with like the safest stem cells for transplantation because, you know, one of the challenges we face, and this is the challenge that we're going after, is how do we keep these cells alive once they're transplanted? But, you know, so it's not necessarily a good thing, but we don't have problems with cell transplantation right now, I think largely because they don't survive. But one of the things we're always worried about is tumor formation. So we really have to make sure that we have a safe population of cells to transplant. And so that's a really important active area that's going on in Andras Naj's lab, for example, to figure out what cells should we be transplanting so that we don't have problems down the road. And so when will it happen? I don't know. But we have seen, there is actually a cell transplantation trial going on right now in spinal cord injury. And that's using embryonic stem cells and differentiating them to these oligodendrocyte progenitor cells and then transplanting that. And that's being pioneered by a company in the United States. So it is happening. It's probably just not happening as fast as we want it to.
Well, thank you very much, Molly. You know, you wonder when you're back in school, when you're learning about the cell membrane, and so forth, what in the world are we going to do with all this stuff? Well, Molly has shown us a wonderful example of taking the content that we learned in school, basic science, and engaging the students in ways. I think this would be a wonderful presentation for a whole lot of people who are teaching music stuff to see where it all goes, how it's all pulled together. Right.
They told me at RSVP, I said, do people need to RSVP and, or can I advertise this at this meeting? And they said, Molly, the room is huge, which just terrified me completely. So I invite you to bring your students or friends, family. It's going to be a different presentation, a little bit more public oriented. I mean, obviously based on science, but because you're all science teachers, a little bit more science oriented. And also, is Joe Grabowski here? No, but he's a high school math teacher in Guelph, and he started an initiative called Exploring by the Seat of Your Pants. And we got in touch with each other through Research to Reality and Exploring by the Seat of Your Pants, and this idea of connecting researchers from across Canada, which is what Research to Reality does, with high school students and middle school students interested in science and math. And so that's an opportunity also, to bring us into your classrooms without literally bringing us into your classrooms, like through a Google Hangout or something like that. Not just me, but there's lots of us.
There is increasingly getting to be an interest. I know about a couple of years ago we had James Hill, who was one of the founding the stem cell work done, and he was just a guest at our luncheon. The teachers went up afterwards and probably was a rock star. He was getting picture taken, the young teachers getting autographs. But the sort of work you're doing, we've somehow got to bring it, lift it up off the page and make it real for our students, whatever. But I want to thank you seriously for the wonderful presentation you did. I think you've given them an awful lot to think about, and maybe go back and say, hey, this is stuff that really is happening and is happening here. Thank you very much.
Thank you.
Now Molly is rushing off to another meeting. I know. What do you think, sir?
Well, it's funny, your first teacher